Um, sorry. Do you want to start with the sponsor slide? Oh, that's fine too. Hi, everyone. So I'm Alice Saragni from UCLA. Thank you, everyone, for joining us today. As people trickle in, just a couple of reminders from the Society of Functional Precision Medicine. So firstly, AACR is coming up, and we have a reception on Monday afternoon, and anyone is welcome to join us. There is also a section focused on functional precision medicine and oncology that's chaired by Tony Letai, and that will be on Tuesday. The detail is there, and you can find all the information about both of these events on our website. I hope some of you can join us for this. That would be fantastic. And also, I hope all of you are members of the society. And if you aren't, please do join us. It's, it's very cheap, and you get nice seminars and a bunch of other advantages. And so, for all of you that are passionate about functional precision medicine, I hope you will be able to join us. And then we have a few sponsors to thank. If you can pull the slide. There you go. Yeah, so we're thankful to, all the sponsors that make these and our reception possible. And thank you all for being here. Our speaker today is Dr. Jason Sicklick, who is an international, world-renowned surgical oncologist and expert in sarcoma and treating, GIST, so gastrointestinal stromal tumors, retroperitoneal sarcomas, as well as liver tumors. He actually got a medical degree from UCLA, and then, did his residency at Johns Hopkins, as well as research at Duke, and finished his fellowship at Memorial. And he's now a professor of surgery and pharmacology at UC San Diego in the School of Medicine. He also is a co-leader of the sarcoma disease team at the UC San Diego, and a member of the NCCN for soft tissue sarcoma, as well for the GIST subcommittee. And so he helps develop really the best practices to treat and handle these rare tumors. He's also an elected member of the American Surgical Association, and his research focus is on conducting clinical research to improve our understanding of complex rare cancers such as GIST and retroperitoneal sarcomas, and really find best precision medicine approaches for treating aggressive metastatic cancers. His laboratory is focused on molecular mechanisms of GIST development, as well as drug resistance in advanced disease. He has been at UC San Diego since 2010. He has co-authored over 130 peer-reviewed articles in prestigious venues, and is on the editorial boards of Cancer, as well as the Journal of Gastrointestinal Surgery. He has received numerous awards for his contribution and to advancing GIST treatment, including in 2021 a Rare Impact Award from the National Organization for Rare Disorders, so from NORD. Today, he will talk to us about moving from drug-centric to patient-centric precision oncology, lesson learned from the iPredict study, and I'm very much looking forward to hear your talk. Thank you so much for being with us, Jason.
Thank you very much, Alice, for the introduction and for the invitation to speak today. Are you able to see those okay?
Yep, perfect. Thank you.
Perfect. So, so as Alice mentioned, I'm going to be speaking about our work in precision medicine. And specifically, we'll talk about the iPredict study that we ran here in San Diego. These are my disclosures. So I think even if you listen to my introduction, you sort of heard that I co-lead our sarcoma disease team, and I've got an interest in GIST. And if we think about diseases, we always are concentrating on the type of disease, and that's really tumor classification based upon disease type has become our religion, if you will. But really, as time is going on, we need to start thinking about this in a different way. We need to start thinking about this in terms of cancer biology, and not necessarily in the realm of a specific disease like GIST or sarcoma or colon cancer, et cetera, because there's so much overlap between the biology of these different tumor types. We obviously know that cancer is a disease of the genome. It's caused by carcinogens, viruses, radiation, mistakes in DNA repair, and all of these can lead to alterations that then ultimately lead to malignancy. And when we see these alterations, we see altered proteins, altered signaling pathways, and ultimately the end result being malignancy. But one of the problems that we all sort of face is the fact that there's intertumor as well as intratumoral heterogeneity, meaning tumor heterogeneity between tumors within the same patient and even within the same tumor. This is data from the TCGA looking at gastric cancer, and you can see even within the locations within the stomach, we would expect that all stomach tumors would be very similar in terms of adenocarcinoma. But the reality is, depending upon where a tumor occurs in the stomach, the molecular profile is unique to that area. If we look at biliary tract cancers, for many years, we've all assumed that biliary tract cancers, whether it be intrahepatic cholangiocarcinoma, extrahepatic cholangiocarcinoma in the extrahepatic bile ducts, or gallbladder cancers, are all the same cell population giving rise to these tumors. And if we think about it histologically, we basically know that there is heterogeneity within the histology of these tumors. But despite this, we sort of have persisted over the years in chemotherapy regimens that are mediocre at best, with median overall survivals you can see there in anywhere from the 5 to 15 month range for gemcitabine cisplatin or 5-FU based treatment regimens. Begging the question is, can we do better for treating these patients? If we look at these, though, not every tumor is the same. What it turns out is that there's a small amount of overlap between the molecular biology of these tumors. However, each of these tumor types has a very unique molecular signature. And so, for instance, if we look at intrahepatic cholangiocarcinoma, and this is where we will frequently see the FGFR or fibroblast growth factor receptor fusions, as well as isocitrate dehydrogenase or IDH1 mutations, BAT mutations. If we look down at the in green there, the gallbladder cancers, these have a completely different molecular profile with the ERB pathway and PTEN, PIK3CA pathway. And then finally, again, if we go over to in blue, the extrahepatic cholangiocarcinomas, again, a different molecular signaling pathways, including ARID1A. So one of the things that, as many of you are aware and obviously involved in this in the precision medicine field, is that we need to be thinking about moving away from sort of just blind shooting targets or shooting arrows at targets with chemotherapy. And we need to be thinking about how we can utilize targeted therapies, in a more biomarker-driven way to target cancers. So the promise of precision oncology was that we could sequence patients, we could stratify, and then we can match them. We can move from basically saying we're gonna take a population of patients, we're gonna treat them in a histology dependent fashion that's agnostic to any biomarker, with a one medicine fits all approach, and try to move towards identifying single marker genes that could theoretically correlate with systemic therapies and treat those patients. And this started out with genetic testing, with single gene marker testing or hotspot testing, where we could identify mutations in individual genes or copy number alterations. But this allowed us only to gain information about a limited number of alterations at one time. These tests sort of needed to be planned a priori and often resulted in exhausted tumor tissue. And this was around this is, when you look back, really, this was only 13 years ago that was sort of where we were in the field. In 2010, Dan von Hoff's group published really what sort of became one of the seminal papers in what we would think about now as current day precision oncology, treating patients in a histology agnostic fashion using RNA and DNA sequencing to guide therapy. And in that publication they published in JCO in 2010, they basically performed biopsies on patients, did immunostaining, FISH, and microarray, and basically said, if we don't find a target, we're going to treat the patients by clinician's choice. But if we do find a target, we're going to give them some type of molecularly matched therapy. Now, one of the endpoint that they sort of used was knowing that with each successive line of therapy, the progression-free survival we would expect to get shorter and shorter as tumor heterogeneity evolves and patients' tumors develop more resistance mechanisms. But they hypothesized that if they could molecularly match a patient and prolong the PFS in this next line of therapy relative to the first line of therapy, that would be a win. And what they said is, if we can do a greater than 30% prolongation of the PFS ratio of the PFS2 to the PFS1, we would take that as a positive signal. And what you can see here in this figure from their paper is basically that a quarter of patients had prolongation of their progression-free survival in their molecularly matched therapy relative to their prior line of therapy. Now, a couple years later, there was the PREDICT study was performed at MD Anderson, and this was sort of a follow-on study and utilized hotspot testing that was available at the time to basically molecularly match patients in their phase 1 unit to match therapies based upon alterations identified in a limited number of genes. And their primary endpoint was basically looking at the clinical benefit rate, meaning the complete responses, the partial responses, and the stable disease for at least 6 months in patients being treated in a phase 1 unit, having failed multiple prior lines of systemic therapy. What you can see there on the left side in the waterfall plot is that those patients that received a molecularly matched therapy had a disease or a clinical benefit rate of 50% versus only 15% on the unmatched group on the right side, suggesting that even with limited numbers of genes, but a broad panel of drugs potentially available, we could see clinical benefit for patients treated with molecularly matched therapies. And so this sort of now starts leading to where can we start moving in the field? Can we start moving towards sort of a more personalized treatments where we really start thinking about the individual patient, utilizing more than just a single gene marker, but histology, multiple gene markers, RNA and DNA sequencing to start thinking about personalizing therapy. So this is now, sort of takes advantage of where we are now in 2023 with next generation sequencing and our ability to identify hundreds of clinically relevant mutations all at once, and allows us to really start, delving into the in-depth biology of patients' tumors while also sparing tissue because of the efficiency of next-generation sequencing. So precision medicine is really now continuing to evolve into a couple of different types of models. One being clinical trials based upon a basket trial or one gene, different histologies. So, whereby we look at patients with this example being a breast cancer, a gastric cancer, and a gallbladder cancer, but they all have biomarker A, and so therefore they would receive a drug targeted against biomarker A. The alternative being umbrella trials where it's one histology but different genes, where, for instance, lung cancer 1, 2, and 3 all have different genomic alterations, and patients would be molecularly matched in this type of clinical trial to the drug that best matches one of their molecular alterations. And you can see there for patient 1, that would be drug A, for patient 2, that would be drug E, and for patient 3, that would be drug G. And it's through these types of trials that we've really made a lot of progress over the last several years. And the FDA has really moved towards, embracing this sort of changes in the paradigm of clinical trials, as well as FDA approvals for targeted, therapies based upon genomic alterations. This list here is a list of the 8 drugs that are now FDA approved in a tumor agnostic genomic based fashion, meaning that independent of the tumor histology, these drugs can be administered to patients if they have the alteration that correlates with the drug for which they are FDA approved, including, for instance, pembrolizumab for microsatellite unstable or DNA mismatch repair deficient. You can see there the larotrectinib or entrectinib for NTRK fusions, all the way on down, including a high tumor mutation burden and several other newer targets. So with this, though, if we look back a little bit, we're certainly seeing benefits of precision targeted therapies. But what we're sort of noticing is, if you start looking at bigger data, is that we're hitting this glass ceiling of about a 30% response rate. This was a study looking at 85,000 patients in phase 1 and phase 2 clinical trials. What you can see there is that patients that received non-precision targeted therapies, meaning that they received a targeted therapy, but they didn't have the actual molecular biomarker for that target, response rates are roughly about 5%. With standard systemic chemotherapy regimens, we're talking about 15% response rates across the board in this large analyses of patients. And in precision targeted therapies, meaning a molecularly matched therapy to a single genomic alteration, these studies would suggest that we're sort of, as I mentioned before, hitting that glass ceiling of about a 30% response rate. So where does this sort of bring us to? Well, there's been lots of large trials done in the United States and as well as around the world. And I'm just going to touch on a couple of them. One of the largest being the NCI MATCH trial that was started in 2015, asking the question is, could we utilize a precision medicine approach in a real-world practice of prescribing targeted therapies outside their approved indications? And this is based upon the data that has been published to date. What you can see here, of the 39 study protocols, there have been 13% of them have been positive study protocols with objective response rates between 11 to 38%. Again, sort of falling, just as I mentioned, sort of in that, kind of peaking out in the 30s in terms of response rates. You can see that about a quarter of the study protocols were negative, and then a majority of them are actually still unknown or ongoing. But, if we look overall, what we see is about 90% of these study protocols were single agent therapies. If we look at the Shiva trial, this was a study done in France. And I think this was a randomized trial with the goal of trying to utilize predefined molecularly targeted therapies versus conventional chemotherapy or best supportive care. The study looked at 741 patients. They all had metastatic disease and had progressed on standard lines of therapy with a median of 3 prior lines of therapy. Patients received broad genomic testing. And of the 741 patients, 40% of patients had at least one molecular alteration identified in pre-specified targets, including hormone receptors, the PI3, AKT, mTOR pathway, RAS, RAF, MEK, ERK pathway, representing molecularly targeted, therapies available for those alterations. And so they went on to assign 195 patients, or 26% of the original cohort, to a targeted therapy. They randomized patients. 99 patients received molecularly targeted therapy, and 92 patients received the physician's choice. And what you can see here is there was no difference in the progression-free survival between the two groups, suggesting, and they basically concluded from their study that the off-label use of molecularly targeted therapies should be discouraged, but enrollment in clinical trials should be encouraged for assessing predictive biomarkers. And so this, I think, really kind of, had a detrimental effect on the precision medicine field and people's view of molecularly matched therapy. And if you go delve into this, there's certainly perhaps you could argue that some of the molecular matches that they treated patients with, including hormone therapy as a single agent against advanced cancers, may have been insufficient to really develop a signal for efficacy in these patients. So what other trials have been done? Well, sort of moving from the 13% positive group or positive study protocols in the NCI match is the ASCO taper study, where they've seen now 28% positive arms. Again, very similar to the NCI match. This was real world practice of prescribing targeted therapies outside their approved indications. You can see here 28% of the study protocols, again, with 39 study protocols have been reported to be positive, 18% negative. And you can see over half are still pending or ongoing as of the latest reports. But what you can also notice that's different, and perhaps one of the explanations that distinguishes the NCI match and the ASCO taper is that in the ASCO taper study, only about a third of protocols are single agent therapies, as opposed to, I mentioned, it was almost 90% in the NCI match. And within the ASCO taper study, 28% include immune checkpoint blockade combinations. And so, this sort of sets the stage for kind of where we are. There's several other trials ongoing right now, including the Target study, which is a liquid biopsy study. There's the Capture study in Canada, the Cupisco in Europe, and the TRACK study in the United States, which is being led by the Target Cancer Foundation to treat patients with rare tumors. But if we sort of take a step back, and this sort of where we kind of fell into this mix is our iPredict study that we started in 2015. And we recognized at the time that there were several challenges to targeted therapy approaches. One is that every tumor is a little bit different, so-called malignant snowflakes, that while they're all, are snowflakes, everyone's a little bit different and everyone has, its unique aspects to it. And the second issue is sort of goes back to some of those early figures I showed you, is that no two tumors are exactly the same in terms of their code genomic alterations. And so, most tumors we recognize are highly heterogeneous. They have spatial heterogeneity. A tumor that arises and metastasizes may be different in the metastasis in different locations. And also tumors change over time. They have temporal heterogeneity as we select for the natural progression of tumors and selection of drug-sensitive clones that become removed from the tumors, and then resistant subclones then are able to proliferate and lead to relapse. And so this brings us then to how can we utilize next generation sequencing, in today's clinical practice. And so, there's really this sort of important aspect of not just having this next generation sequencing, but being able to implement it. And that's where the multidisciplinary approach is critical to this, and trying to essentially crowdsource knowledge. Not only knowledge of sort of the classical things that we think about, the imaging data, the pathologic data, the patient and tumor characteristics, but including in now the molecular information. So, I would say the future is now. We now have molecular tumor boards, across the United States and across the world that integrate this data from patients' tumors in order to begin better interpreting the data and making clinical decisions. And so this isn't just a one person sitting in a room looking at a molecular alteration and deciding, what's the best way to treat this, but really crowdsourcing again and bringing together people from all different disciplines, from the medical, surgical, and radiation oncology side, the radiology and pathology side. But if you look on the right side there, people that have expertise in clinical genetics and germline alterations, bioinformatics experts, as well as molecular biology experts, all sort of bringing this together to provide the best care for patients. And so what does this lead to? Well, data published from our institution showed that molecular tumor boards, have several benefits, including leading to increasing molecular profiling of patients, increased assignment of patients to clinical trials, and then finally receiving more precise molecularly targeted therapies based upon these recommendations. So how do we bring this together, and how do we really start thinking about, rather than drug-centric being that we're going to give one drug because everybody has molecular alteration A, to a more patient-centric, where we can start doing an N of one approach, where we start thinking about patients on the individual level and start thinking about how we can target patient one who's got biomarkers A, B, and C with perhaps drugs A, B, and C versus patient two with drugs A, E, and F. And so this leads to the iPredict study that we began writing this in 2014, and it was ultimately IRB approved and activated in 2015. iPredict was the investigation of profile-related evidence determining individualized cancer therapy, and I co-led this with Roselle Kurzrock, who at the time was at UC San Diego. She's now professor at Medical College of Wisconsin, as well as the chief medical officer of the WIN Consortium, which is the Worldwide Innovation Network. And what we said at the time was, this is now sort of in planning stages, now 9 years ago when we started working on this, was we're going to take patients with highly lethal malignancies, which we define as a greater than 50% two-year mortality, and we're going to attempt to try to treat them with customized combination therapies. And so in the first cohort of patients that we treated, we started enrollment in 2015. In the first cohort, we consented 149 patients, and we treated 83 of these patients, and we molecularly matched 73 of them, representing 88% of the treated patients, or 49% of the entire cohort. This was done in collaboration with Foundation Medicine, utilizing their Foundation One panel, which early on was, had a limited but large number of genomic alterations in their gene sets. But the way we wrote the clinical trial was that it could be very fluid in its adaptation of tumor mutation burden, microsatellite instability, immunostaining for PD-1, PD-L1, circulating tumor DNA, as these types of tests came on to CLIA-approved market. And so basically, as the technology evolved over the course of the trial, we adopted that technology in real time in order to help with our decision making. And so the way patients were this was treated, we were going to take patients that had failed at least one line of therapy and had metastatic or unresectable disease. The patients would be consented. They would undergo genomic profiling by Foundation Medicine. If they had no genomic alterations identified on the right side, they would be treated by the physician's treatment of choice using traditional systemic therapy. But if they had an alteration identified, they would be discussed at our molecular tumor board and via our study committee, and we would recommend molecularly matched therapies. Now, ultimately, while we recommended these therapies, it was ultimately up to the treating physician to make the final treatment choices regarding whether they wanted to treat these patients with molecularly matched therapy or unmatched therapy. What you can see here on the left side is the patient characteristics. Median age was 62. There was two-thirds of patients were women, and you can see there that a majority of the diseases were gastrointestinal and hepatobiliary, with about 42%. GYN was 17%, about 14% breast, and so forth. Noting that, while we have we were learning at the time a lot of information about lung cancer, we made a conscious effort really to minimize the number of patients with lung cancer that we enrolled on the trial, with the expectation that we didn't want to prove that, for instance, erlotinib worked for EGFR mutations in lung cancer. We wanted to go after other aggressive diseases where we didn't have as well-defined molecular targets or molecularly matched therapies. These patients had received a median of 2 prior lines of therapy with a range between 1 to 3. They had a median of 5 genomic alterations with a range between 1 to 19, really speaking to the fact that these patients don't just have one genomic alteration that needs to be treated with one drug, that they have lots of genomic alterations, whether they all be drivers versus a mix of drivers and passengers. That is certainly an area of point of discussion. The patients received a median of 2 drugs on the trial with a range between 1 and 5. And what you can see there on the right side is the different pathways that were targeted, including MAP kinase, immune checkpoint, P53, based on an accumulating amount of data suggesting that P53 is upstream of VEGF, and that alterations in P53 correlate with targeting VEGF. And you can see there all the way down beyond is ERB pathway, PIK3CA, FGF, et cetera. One of the things that we utilized during this trial was a molecular matching score. And what we mean by that is, this is a patient with gallbladder cancer. This was actually the first patient we enrolled on the clinical trial. And if this patient had a BRCA2 mutation, which we would predict would be sensitize them to a platinum-based agent, and if this is a gallbladder cancer, as I mentioned earlier, we could treat those patients with gemcitabine and cisplatin. But this patient also had a PIK3R1 and a PTEN alteration. So the patient had 3 genomic alterations, but if we were to give them patient standard of care, we would say that we targeted 1 out of their 3 alterations, or a 33% molecular match. But if we were to say, if we're going to give them a platinum agent with an agent that could target their PI3 AKT mTOR pathway, we would say that we would have a 100% match, or a 3 out of 3 match. And so you can see on the bottom there, the number of alterations targeted is in the numerator, and the total number of alterations is the denominator, and that was what we used to create our matching score. And so here's the actual data from the trial. If we look at the patients that were treated with molecularly matched therapies, those patients that had at least a 50% molecular matching score, shown there in the bar graph on the left, their partial responses and complete responses in dark green, and their stable disease for at least 6 months being their disease control rate, was 50% in those patients that received high molecular matching scores, versus only 22% in those patients with lower molecular matching scores. On the right, if we go back to the Von Hoff PFS ratio approach, if you look at those patients that had high molecular matching scores, there was 75% of patients had prolongation of their progression-free survival by greater than 30% in the on-trial treatment relative to their prior line of therapy, versus those patients with the low molecular matching scores, only 37% had prolongation of their PFS in the next line of therapy. So what does this mean in terms of survival outcomes? Well, if you look on the top there, this is the progression-free survival. The median PFS for the high molecular matching group was 6.5 months versus 3.1 months in the low molecular matching group, with a hazard ratio of 0.4. And at the time, this on the bottom right there, this was the first study that I was aware of that showed a difference in the overall survival, not based upon treating patients with a drug specific for a target, but based upon a molecular targeted approach of just utilizing an algorithm, if you will, or knowledge in order to treat patients. And what you can see there is that the high molecular matching score group, the median overall survival was not reached, versus it was 10 months in the low molecular matching score group, with a hazard ratio of 0.44 and a p-value of 0.46. So, this was really, sort of early evidence that we could start giving customized combination therapies together. If you look in the publication that we published in Nature Medicine, we have a lot of data also on the safety data of it, showing that there was no differences in the safety profiles between the low or high or unmatched groups. But, what about where are we in the real world? And so we went back to our data at UC San Diego and looked at our real world molecular tumor board data. And this was a project led by Shumei Kato, one of my partners. And we looked at all patients presented at our molecular tumor board with the emphasis on giving patients customized combination therapies. The period of this study was from 2012 to 2018. It's included 200 molecular tumor boards, encompassing 715 patients and 58 physicians that have presented at least one or more cases. We included any treatment decisions that could be guided by any kind of testing that was done. And so with these 715 patients, the median age was again in the 60s. And again, there was a predilection towards women. You can see there, not surprisingly, just based upon the frequency of different tumor types, the most common histologies included breast cancer, colon cancer, hematologic malignancies, et cetera. If we look at the different tumor types and tumor testing assays, you can see it was really mixed. We used tissue next generation sequencing. You can see there was predominantly Foundation 1 with about 20% of other tests at the time. Circulating tumor DNA, including Garden and Foundation ACT, and then immunotherapy biomarkers, including the Foundation Medicines panel, as well as others. And what we saw is very similar. If we looked at the high molecular matching group, we saw an improved disease control rates on the bar graphs on the left there in green, with complete responses, partial responses, and stable disease for greater than 6 months, versus lower rates on the right side. If we look at the overall survival, what you can see there is in patients that had a high molecular matching score from 50 to 100 versus those in green versus those with 0 to 49%. You can see also a difference in the overall survival between these patients. But what's probably was very quite interesting from this study, which was sort of added, a new concept to this, was that higher compliance with the molecular tumor board recommendations resulted in better outcomes. If the treating physicians followed all recommendations in green versus some of the recommendations in blue versus none of the recommendations in red, you can see there that there was differences in the progression-free survival on the left side and the overall survival on the right side, suggesting that the importance of molecular tumor boards, of discussing patients, of working together as a team in order to provide the best care for our patients. But this begs the question is, can we still do better? Can we improve upon the iPredict customized combination therapies, where I showed you that we may have sort of surpassed that glass ceiling of 30% response rates of molecularly matched therapies, and getting this up to the 40 to 50% range, and advance beyond the knowledge from combining and crowdsourcing knowledge within a molecular tumor board to surpass this 50% disease control rate. And so as part of the iPredict study, we asked the question is, can we sort of preempt the temporal heterogeneity and the tumor evolution that occurs with treatment? Could we think about treating patients in the first line before their tumor heterogeneity involves? And so this was an added on arm of the iPredict trial, where we said, we're going to treat patients with first line customized combination therapies and forego the standard of care therapies. And as you can imagine, this did not go over well early on with the IRB, and it took us well over a year to ultimately get the trial approved by the IRB before we could get this open to enrollment. But like the other arm that was the previously treated arm that I just discussed, we consented 145 patients. We treated 52% of them, including molecularly matched therapy in 71% of these patients. And again, this was using the Foundation Medicine panels. Very similar demographics, mid-60s or mid to early 60s, slightly more women than men, higher rates of non-colorectal GI malignancies, as well as carcinomas of unknown primary, which were often cholangiocarcinomas, hepatobiliary tumors, and so forth. Like the patients that were previously treated, there was a median of 5 genomic alterations per patient, but some patients had no genomic alterations identified, as opposed to every patient in the previously treated arm had at least one. And in this group, we treated 80% of patients with at least 2 drugs versus about 20% with a single agent. And you can see there on the right side, in the bar graph, the types of genomic alterations that were targeted in these patients. This is a combination of a waterfall plot as well as swimmer's plot for each of these patients based upon the histology. You can see there bottom panel based upon the colors. And on the right side here is this combined waterfall swimmers plot, where what you can see there on the left side is those patients in red or pink with low molecular matching scores tended to cluster to the left side here and tended to have either stable disease or early progression with short durations of treatment, versus on the right side here, you can see that the patients that had more deep responses, as well as were maintained on treatment longer, tended to have higher molecular matching rates. And so when we went back and we sort of analyzed the data in this cohort, it actually turned out that there was sort of the matching score cutoff was slightly higher at 60%. So we said we're going to look at patients with a higher matching score greater than 60%. And of those patients, 68% had the disease control rate was 68% here on the left side versus 38% on the right side. Now, if we broke this data up further and pulled out those patients that had unmatched therapies, meaning the molecular matching score was 0, which you can see here, we can now move from a 68% to 39% to 20% disease control rates, suggesting that molecular matching score in the treatment naive setting does correlate with the response rates, and that we could start surpassing even that 30 or even that 50% rate of disease control and getting patients up to the 70% disease control rate. If you look on the right side there, this is the Kaplan-Meier curves, the PFS. You can see a difference of about 9 months between the high versus the low molecular matching group. And on the lower panel there, this is the overall survival. You see about a 7-month difference with a hazard ratio of 0.51 and a p-value, you can see there, nearing significance with a p-value of 0.053. But probably the most interesting part of the analyses that we did was that we binned each patient into different groups. So we said if they're either a 0 or unmatched, they're in the 1 to 39%, 40 to 59%, 60 to 99, or a perfect 100% match. And what you can see there is that when we look at these binned groups and their response rates, or their disease control rates, or their median progression-free survivals on the y-axis, you can see it's nearly a perfect linear correlation with R-squared values between 0.92 and 0.99 for on the left and right there, as you can see with the red boxes. That molecular, this is not a black and white, all or none, matched or unmatched. This is about, this is a gradient, and that the better we can match patients, the better responses that we tend to see in these patients. And so, what have we learned from this? Well, we've learned a lot of things, in the course of this trial. One is that DNA and RNA and immunostaining all together need to be brought in as part of the precision medicine approach. That single agents are often ineffective to treat lethal cancers, that we need a multidisciplinary approach to bring the best treatment for our patients, that we can start thinking about, bringing in targeted therapies combined with immunotherapies. And I'll talk about that in a couple of minutes with some examples, that we can safely treat these patients. And again, for the sake of time, in this latest trial arm that we published, there was no difference. And in fact, actually the higher molecular matching group tended to have lower adverse event rates than the lower molecular matching group. And ultimately, we're trying to bring this all together to personalize precision medicine approaches for our patients. So I'll just give you a couple examples of cases that we treated on the trial. This was a patient with a stage 4 intrahepatic cholangiocarcinoma. You can see there had the KRAS alterations and MLH1 and P53 alteration. And the patient was administered trametinib to target the KRAS and nivolumab for the MLH1 alteration. And what you can see here on the CT scans is this lesion up in the left side of the liver, as well as this portocaval node shrunk down. And you can see here even further response by 52 weeks and a dramatic decrease in the CA19-9 tumor marker. Here's that original gallbladder cancer patient that I mentioned in the original description. We treated her with carboplatin and everolimus, and you can see here between the left side and the right side, after just an 8-week period, a dramatic response to therapy in the liver as well as outside the liver. Now, unfortunately, this speaks to the fact there is tumor heterogeneity in these patients. And while, we saw dramatic responses to these tumors that we knew the molecular alterations of, she developed new disease here in the backside of the right side of the liver, within that short period of time. Suggesting that, even though we're targeting these patients, it really speaks to the fact there is heterogeneity that we still don't appreciate. Here's a pancreas adenocarcinoma patient who had a KRAS and ERB2 and a CDK N2A alterations. This patient had a 50% molecular matching score, where we targeted the ERB2 with trastuzumab, as well as lapatinib, the KRAS with trametinib, and the CDK alteration with a CDK4/6 inhibitor. And what you can see here at week 0, you can see, diffuse metastatic disease, including lung mets. And after just 4 weeks of treatment, you can see responses across these lesions. Probably one of the more dramatic examples is this was a patient that presented to me. I don't, as Alice mentioned, I generally take care of sarcomas, but she was sent to me with a stage 4 triple negative breast cancer with a bowel obstruction, and the referring physician just asked me if I could divert her to send her to hospice, as she'd failed 6 prior lines of therapy, including 10 different drugs. She had peritoneal disease, she had retroperitoneal nodes, her ureter was blocked up, causing hydronephrosis, she had ascites. And so I took her to the operating room. I did a laparoscopy on her, identified peritoneal disease, did her diversion, and it turned out that we sent off her tumor for molecular testing, and her tumor mutation burden was 76. And disregard this dabrafenib, that's an error I'm just noticing. Her tumor mutation burden was 76, and this was very early on in the days of understanding tumor mutation burden and responses to therapy, because remember, now we're talking 2016. And so, but we did know that higher tumor mutation burden was associated with higher levels of tumor neoantigens, as was originally described by Lyudmila Alexandrov, who I have the pleasure of working with. He's just 1 floor below me here in the cancer center. And so we knew that these patients had higher neoantigens. And there was data published around that time demonstrating that that patients with high tumor mutation burden had higher response rates to immune checkpoint blockade. And this was before the Keynote 158 study, which ultimately led to pembrolizumab being FDA approved for patients with a high tumor mutation burden. And so we consented her for the iPredict study. We put her on nivolumab. We started her on treatment in 2017. And she had a dramatic response to therapy. And I'm just going to stop sharing here for 1 sec. And I just want to share this quick video with you from her.
2016 promised to be a year of milestones and celebrations. All 3 of our children were graduating that year. Our 2 daughters were graduating from college, and our son was graduating from high school. I went in to a routine oncology appointment. It was anything but routine. My tumor markers were highly elevated, and there was little doubt that I had metastatic breast cancer. Over the next 7 months, I failed numerous lines of chemotherapy. The year that had begun with so much hope and promise really looked like my last full year of life. I tried to condense a lifetime of a mother's love and guidance and wishes into each child's journal entry. Lastly, I made peace with God And I was really ready at that point to die because that's where things looked like they were going.
I met Teresa in late 2016. At that point, she was referred to me with a bowel obstruction, and the referring medical oncologist asked me to see her to see if we could undo the blockage so that she could eat, with the goal of sending her to hospice. But at least if we could have her eating, she'd have some comfort and some quality of life before she passed away.
After surgery, I asked Dr. Sicklick if there might be a clinical trial that he would suggest for me. And he recommended the iPredict clinical trial. My oncologist, Dr. Shumi Kato, selected a therapy that was perfectly matched to my cancer. And what follows is just the most remarkable story. After 2 infusions, my tumor markers dropped by 75%. After 8 weeks, my CT scan suggested a complete response to therapy.
Days, weeks are the difference between life and death now.
And so, that was 7 years ago now, or almost 7 years ago. She's still alive and without evidence of disease. She's now gone on to see not only her kids graduate from high school, but she's got grandkids now and is doing quite well. So really, it's a great story of using molecularly matched therapy and precision medicine, in order to treat patients. But we're really just at the tip of the iceberg. All the things I've talked about today are mostly about the DNA, with very little mention actually of the transcriptome. But there's so much more below the surface that we need to be thinking about. As you can see there, the proteome, the epigenome, the metabolome, the microbiome, using other biomarkers and artificial intelligence, all in order to treat our patients. And with that, I want to finish and say thank you to all of you for your attention, and thank you to the patients and their families for trusting their care in us, and happy to answer any questions. Thank you.
Thank you. Thank you so much, Jason. This was a fantastic talk and really, wonderful excursion into the history of genomic precision medicine. And so if anyone has a question, feel free to raise your hand, or if you don't want, you can just, drop it in the chat. Maybe I'll get us started. And, of course, for those patients who you match to a high degree, but still don't fare well, do you actually have any sense as to whether this is due to higher heterogeneity, whether, intralesion or across lesion? Is it higher clonal diversity? Is it lower allele frequency? Are you looking into that? Do you have any sense of what's happening?
Yeah, no, I think it's probably all those things. Do I have definitive data on that? No, unfortunately, as part of the trial, one of the things we did not do was to do post-treatment biopsies on these patients. And so, I can't, speak to that. We do know that, just from looking at our data across the cancer center over time and looking at, some of these subset analyses that we've done and published, is that with, each lines of therapy, we're seeing that patients are progressively getting more, genomic alterations in their tumors that are, leading to new resistance mechanisms. And so I certainly think it's a problem. Along those lines, one of the other issues is that, not every disease sheds ctDNA. And so while, a lot of people, like lung cancer is a great example where it does, but there's a lot of diseases where it does not. And so, while there's been a tremendous number of retrospective studies and there's prospective studies all trying to say, can we use ctDNA, as an adjunct in order to understand tumor evolution and tumor heterogeneity, there still is more we need to understand of why certain cancers are shedding and some are not shedding, as well, in order to understand that clonal heterogeneity as well.
Thank you. And we have a question from Mark Stevens. Do you want to say that out loud? Otherwise I can read it. All right, I'll maybe read it. Okay, so he's saying the outcome distribution and correlation you've shown has been by matching score percentage are interesting. How do those distribution look when been by number of therapies used for treatment? So 1, 2, 3, 4 plus. Or alternatively, what fraction of the correlation is made up by number of drugs received?
Great question. So, so we did in the publications, I don't remember the exact specific statistics off the top of my head, but in the multivariate analysis, the patients that received 2 or more drugs, that was an independent predictor of response as well. So speaking to your question or your there. So alternatively, what fraction? I don't, off the top of my head, I don't know the fraction of correlation made up by the number of drugs received per se. It's something that we'd have to go back post hoc and look at. We've closed the trial to enrollment. We set last December 1st, we set the data cutoff date for collecting data, and we're now doing a lot of post hoc analyses of the data. These are great questions for us to look at. Ultimately, by the time we closed the trial to enrollment, we'd enrolled 505 patients on the trial, and we treated about 270 of them. So there's still a wealth of additional data for us to mine from these patients, including,
sorry, go ahead.
Sorry, including some of the questions that you're asking that we now may have more power to answer some of these with larger numbers of patients.
And the last part of the question is whether, the results you're getting are actually, I guess, biased toward outcomes of combination with checkpoint inhibitors.
Yeah, no, I mean, I think, it's another good question. I mean, we certainly use checkpoint inhibitors as an adjunct, within, but it was, in situations outside of high, tumor mutation burden, which you can see like that patient was well before the FDA approval, or, we were even thinking about giving somebody, immune checkpoint blockade with a high tumor mutation burden, or it was just kind of, starting to scratch the surface of it. We did utilize that, but often in combination as appropriate. For her, she had, I think it was 12 different genomic alterations. There's, no physical way to give 12 different chemotherapeutic agents to target all those. But, this was a great opportunity for her to be treated with immune checkpoint blockade. But often we did do it in combination. Another good example was that patient with the intrahepatic cholangiocarcinoma. I mean, we know response rates for intrahepatic cholangiocarcinoma, predating the FGFR inhibitors, which still aren't great, were incredibly low. And so, that for that patient to have a near radiologic response is pretty dramatic with the combination.
Hi, this is Chris Kemp calling. I have a quick, that was beautiful. Thank you so much. And especially for sharing the patient story. So I see on your iceberg, I didn't see a role for functional testing, which of course, this is the Society of Functional Precision Medicine. And so I just want to make a quick comment that in our analysis of hundreds of patients, yes, some have genomic alterations which you can target, but some don't, of course. What we found is that those who don't almost universally are vulnerable to targeted therapies nonetheless. We don't know what the biomarker is. And when patients, we have case studies of patients taking such drugs without any known genomic biomarker and responding with long-term tumor regression. So I just want to make that as a comment going forward. And then I have a question, which I asked Rizal when she gave a talk at the same group like last year. How do you go about obtaining off-label drugs for your patients, and who pays for them?
So first off, your comment is great, and I totally agree with it. It's it's a challenge to functionally understand some of these patients, and to try to even do this in real time with the patients, where we could functionally do this and try it in real time to pick drugs or combinations of drugs that might work for a given patient in real time. But I absolutely think it's an important part of doing this. We'd actually, at one point, there was a company that had an assay that ultimately they went out of business, that we were going to try to work with. To the next iteration of the trial was going to be called CanPredict, and it was actually going to use a type of functional approach that you're describing. Clearly, the company went out of business, so their approach didn't ultimately pan out in the end. But that was going to be part of it. In terms of obtaining the drugs, it's an important factor. We actually have somebody that is full-time employed here. We've deemed her a medication acquisition specialist whose entire job is to focus on getting drugs for patients. We all know that getting approvals for drugs and working with the insurance companies can be incredibly difficult. Sometimes, and we're all busy clinicians. You've got time to make one phone call to an insurance company to try to advocate for your patient, but beyond that, it becomes quite difficult. Her entire job is doing that, and whether it be getting the drug, through approval through the insurance company after letters and sort of arguing the case for it, or even working with drug companies, and that the lady that I showed you with the nivolumab, it was actually the insurance company declined the approval of that drug for her. And when it came to literally at day 365 of her being treated on the drug, the drug company said that had been actually administering or giving the drug for free, said, hey, we're not giving any more drug. We've proven that it's worked, for your patient. The insurance company needs to take this over. And the following day, the insurance company took it over and, started covering drug for that patient. So that was now 2018. So we're talking 5 years ago now that happened. But, it's certainly been an evolution. But I think it's also, as the insurance companies are, the mindset of precision medicine, is evolving. It's certainly going to help make things easier for us as time goes on.
Thank you so much, Jason.
Maybe one last question, Pamela?
Thank you. That was a terrific overview of the field. It's really impressive. Thank you. What I wanted to mention is along the lines of the insurance authorization. So most of my work has been in heme malignancies, but I was really surprised so we've been asking for insurance clearance for a long time. And initially we were confining our work to where there at least had been some clinical use in that indication for the drugs that we were choosing for patients, largely based on functional screening, drug screening, as well as some molecular targeting. But more recently, I was really surprised that our folks asking for financial clearance, City of Hope went ahead and said that the drug was in a functional screen, and they did approve the regimen. The patient went into remission, went from like 93% blast to 0% blast in the first cycle. And then when I went back and asked for the second cycle or tried to get the drug, we couldn't get the drug for home use. Like we couldn't get the prescription coverage the first time. But when I said the patient went into remission with this, not only do we have the data, but the patient went into remission, they did eventually on peer-to-peer, they did approve the even prescribing a subcutaneous injection with this chemotherapy drug. So I think that as we move forward, this never would have happened 5 years ago or longer, but things are changing. And I've been pretty impressed now that we can, use our own data to try to get the drugs approved. And I just wanted to share that. And again, thank you for your fabulous talk. We did once screen a cholangiocarcinoma in our drug screen, and it was very informative information too. So I have we've gone into some solid tumors, but so I really respect what you've done. Thank you.
Thank you so much. And that's phenomenal to hear that story. And it's really a testament to the way that the field is changing. And, hopefully the insurance, companies are, changing to some degree. That's probably going to be a slower process than as you all move the field forward. But certainly the more data we can accumulate and the more N of one, albeit back, we would sort of, look down on a given case report of something working for a given patient. But as we understand more about the biology, lots of case reports altogether become actually evidence that an approach actually might work, or a drug might, be utilized for given patients. And so I think, it's important to continue pushing this forward as a field.
So thank you so much. Thank you, Jason, for the wonderful talk, and thanks everyone for the great discussion. I appreciate everyone staying on. If you are at AACR, please do remember to join us on Monday. That would be great to see you all there, and see you in a month otherwise. Thank you so much. Bye-bye.
Thank you very much. Have a great day.